Okay, hi everyone, and this is another video. This time around, we'll be talking about Module 3.1 and 3.2, which is cellular respiration. For our purpose of our discussion, we'll just be concentrating on how glucose, carbohydrates will be converted into energy through cellular respiration. So for other food products like proteins and oils and lipids, which can also be converted into energy through cellular respiration, this will not be dealt over here in our discussion, but rather it will be dealt in your higher biology. Now, do take note as well that the photosynthesis process will be the one producing the carbohydrate sugar, which we will then be eating them up and digested through the process of cellular respiration. So let's start with describing cellular respiration. Here are two questions, starting with the first one. Is cellular respiration anabolic or catabolic? And probably you're very familiar with this already. Let's define the two terms. When we talk about catabolism, it is catabolic. It means breakdown of larger molecules. In this case, one example would be proteins being broken down into their constituent amino acid. On the other hand, anabolism is described as, or it is defined as, synthesis of larger molecules. From our example, in this case, amino acid would now be put together to produce the larger protein. So that's the difference between catabolism and anabolism. So let's go back to our question. Is cellular respiration anabolic or catabolic? The answer to this is it is catabolic. So you're very familiar with this. So how about the second question? Is it exergonic or endergonic? Of course, we have to define it first. When we talk about exergonic reaction, it means it proceeds with a net release of energy and is spontaneous. In other words, energy is produced by the reaction. On the other hand, when we talk about endergonic reaction, it means it absorbs free energy from its surrounding and is non-spontaneous. In other words, energy is needed, energy is absorbed by the system in order for the reaction to occur. So going back to our question, is cellular respiration exergonic or endergonic? Well, you know the answer. The answer is it is exergonic. So with that, let's now describe cellular respiration. So cellular respiration is both catabolic and also exergonic. And when we define cellular respiration, it is the breaking down of organic molecules to produce ATP or adenosine triphosphate. So very familiar. Now there are two more terms you have to take note of, the word aerobic and anaerobic respiration. And in order to define this, what is involved with aerobic and anaerobic respiration? Now you're very familiar as well. It is related with the presence or absence of oxygen. So when we talk about aerobic respiration, this would be the presence of oxygen, while anaerobic respiration would be the absence of oxygen. So one example would be the normal breathing that we have. This is of course with oxygen, and it's considered to be aerobic respiration. But when you are running a marathon, you're at the end of your strength and gasping for air, you don't have enough air breathing, you're not breathing enough air while you're running, that is considered to be anaerobic respiration because your muscles are undergoing respiration, but since there is not enough oxygen, that would be anaerobic respiration. All right, so again, we have to take note that cellular respiration is in an oxidation reduction reaction. So that's one important description of cellular respiration again. Particularly, it is the glucose that is oxidized to become carbon dioxide while the oxygen is reduced to produce water. So do take note, cellular respiration is an oxidation reduction reaction. So the glucose is oxidized to carbon dioxide and oxygen is reduced to water. You're very familiar as well with this, the mnemonics, Leora versus Geroa. So Leora loses electrons, oxidize, reducing agent. Gains electrons, reduce, oxidizing agent. So this will be very important thing to remember about cellular respiration being an oxidation reduction process. Now, another description that is very important about cellular respiration is that it releases energy small bits at a time. As you can see in the diagram on the right, you will see here that eventually, we'll be dealing with this a little bit later, eventually the electron transport chain involved in cellular respiration will release energy one bit at a time, releasing ATPs one bit at a time as well. So you can see that cellular respiration produces little bits of energy at a time. However, compared with the uncontrolled reaction, it is an explosive release of heat and light. And imagine your food, for example, a piece of bread or your potato that you're eating. You release all of the energy at the same time. It will be very explosive, just like a car exploding. It's no different from your car. You put 5 to 7 gallons, liters of fuel, and then you put a match into the fuel container, and then all at the same time the energy is released, there will be a big explosion. So it's a good thing it doesn't happen to us, because when we eat our bread, we eat our potato, we eat any food that we have, the energy is not released at the same time. If it is released at the same time, our stomach will explode. So it's a good thing that the food we eat is released small bits at a time to produce number of ATPs. So just take note of that. Okay, another thing we have to take note is the structure of ATP. Will not be talking about this too long because you know this already. So ATP has 3 phosphate groups attached to a ribose and to an adenine. So if you have only one phosphate, it's called adenosine monophosphate. If you have 2 phosphate groups, it's called adenosine triphosphate. If you have 3 phosphate groups, it is called adenosine triphosphate. And the energy can be found in the bonds of between the first and second phosphate and the second and the first phosphate. So these are high energy. If it's broken down, it releases that energy. So that's ATP. So let's continue on. Here's a question: in which part of the cell does glycolysis takes place? So you have to take note of this. This is very important. It happens not in the mitochondria, but it happens in the cytosol or the cytoplasm. So looking at this diagram, you will see here the 4 important processes, 3 of which are major, and the one is an intermediate or intermediate phase. So you have the glycolysis, a very important process in cellular respiration. You have the pyruvate oxidation, which is in between 2 of the major processes. The second major process, the citric acid cycle, and the third one is the oxidative phosphorylation. Now, please do take note that in order for us to truly understand about the process of cellular respiration, you have to, number 1, look at the carbon number of the molecules. We'll be looking at that a little bit later. Next is, to truly understand, you have to take note, our objective is to be able to count the number of ATPs produced. The third thing you have to take note is that where and how NADH and FADH2 will be produced. So those are 3 important things to take note of as we continue our discussion. The number of carbon in the molecule, how and where NADH and FADH produce, and how and where ATPs would be produced. Okay, let's look at the details here. So for glycolysis, you produce 2 ATPs here and 2 NADH. We'll be looking at the details a little bit later. The glycolysis produces or converts glucose into 2-pyruvate. We'll be looking here at the number of carbons later. The 2-pyruvate molecule will now be converted to 2-acetyl-CoA through the pyruvate oxidation, and in this case 2 NADH will be produced. So in glycolysis, 2 ATP, 2 NADH produced. In pyruvate oxidation, 2 NADH will be produced. In the citric acid cycle, 2 ATPs will be produced while producing 6 NADH and 2 FADH as well. So those are the products, high energy products that will be produced by each of the process. Do take note that the NADH and the FADH will eventually find themselves to be going through the oxidative phosphorylation, electron transport chain, and chemiosmosis, eventually producing more ATP. So to take note, the 2 ATP in the glycolysis and 2 ATP in the citric acid cycle are just a few of them, but most of the ATP is produced through the oxidative phosphorylation around 26 or 28. So a grand total of about 30 or 32 ATPs. And that leads us to the question, how are these ATPs produced? How are these NADH produced? How are these FADH2 produced? We'll look into the details right now. Let's dive first into the glycolysis, and this is the first process we need to take note of. Again, what are the carbon numbers? Where is ATP produced? Where is NADH produced? Starting with that, glucose is a 6-carbon molecule. Now do take note that glycolysis has two important stages or phases. Energy investment phase, and the second one is energy payoff phase. We will see a little bit later. And you're very familiar with the word investment, because in investment you are going to shell out a little amount of money, expecting that there will be a payoff wherein you will be receiving a greater amount of money. And that's no different from glycolysis, because in glycolysis you have to invest ATPs, a small amount, but later you will see that there will be a payoff of a greater amount of ATPs. So starting with glucose, which is 6-carbon, the objective of the glycolysis is to eventually reduce this into a 3-carbon molecule, which means glucose will be essentially cut in half. But in order to produce that, 2 ATPs must be invested to cut the glucose into 2 copies of 3-carbon molecules. So that's the objective of glycolysis, from 6-carbon to make it 3-carbon. But it's not yet done, because we have the energy payoff phase, wherein the 3-carbon intermediate will now be converted into pyruvate, which is also 3-carbon. But the conversion of the 3-carbon intermediate into pyruvate will produce 4 ATPs and 2 NADH. And that is where the NADH and ATP will be coming from. Now do take note, you invested 2 ATPs, you gained 4 ATPs, which means you have a net of 2 ATP. So from 6-carbon, it is now converted into a 3-carbon pyruvate with the production of a net of 2 ATPs and a net of 2 NADH. So here is what it looks like. Since there are 2 molecules of pyruvate, so the first molecule will produce 2 ATPs, 1 NADH, and the other molecule will produce 2 ATPs and 1 NADH. So with the investment of 2 ATP, you minus this from the 1, 2, 3, 4 produced. So there is 2 pyruvate, 2 ATP, and 2 NADH produced. And that's it about glycolysis. So very easy to remember here. So what happens next? Now before we continue, just do take note that this is where the 2 ATPs will be coming from and the 2 NADH produced. So we have explained that a little while ago. Okay, now let's continue with the next part, which is the pyruvate oxidation. So in this case, look again at the carbon. The pyruvate, which is a 3-carbon molecule, will now be converted into acetyl-CoA, which is a 2-carbon molecule, which means one of the carbon was released. And how is it released? It's released as carbon dioxide. So it is in the pyruvate oxidation that the first few of the carbon dioxide would be released. Now do take note, no ATP is produced, but rather NADH will be produced. So let's look at the details of this one. But before that, it's the linking step between glycolysis and Krebs cycle. It is where pyruvate is transported to the mitochondria, from the cytosol to the mitochondria, and the pyruvate dicarboxylate is the name of the enzyme, very important for pyruvate oxidation. So let's look again at the details. So you have 1 copy of 3-carbon pyruvate releasing 1 carbon dioxide, thereby producing here a 2-carbon acetyl-CoA, but with the release of NADH over here. So carbon dioxide and 1 NADH produced. But since there are 2 copies of the pyruvate, then you will produce 2 NADH, 2 carbon dioxide, and 2 of that acetyl-CoA, which is a 2-carbon molecule. And that's it for the intermediate process, which is oxidation, pyruvate oxidation. So it's quite easy to remember. So going back here to our drawing, we now know where the 2 ATPs were produced and the 2 NADH produced in glycolysis to produce 2-pyruvate. And then the 2-pyruvate is converted into 2-acetyl-CoA with the production of 2 NADH. Okay, so now let's go straight into the citric acid cycle. So we'll see the ATP, the NADH, and the FADH2 produced here. Okay, so as the acetyl-CoA enters into the Krebs cycle, we won't be looking at the details of the intermediate molecule, but simply look at the carbon number, simply look where the FADH2 is produced, the ATP is produced, and the NADH is produced. So the acetyl-CoA will combine with the 4-carbon molecule of the cycle to produce a 6-carbon molecule. From there, it will now be converted into several intermediate forms, which essentially becomes 4-carbon again. So notice that the 6-carbon becomes 4-carbon, regenerating this, but it now lacks 2 carbon. So where did the 2 carbon go to? It now becomes released as carbon dioxide. So this is the second place where carbon dioxide is produced. The first being the pyruvate oxidation, and the second one being the Krebs cycle. Now, not only is carbon dioxide produced, but the conversion into the 4-carbon produces or releases 3 NADH and 1 FADH or 2. Not only that, it produces 1 ATP. So 1 acetyl-CoA produces 2 carbon dioxide, 3 NADH, 1 ATP, and 1 FADH2. Now please do take note that there are 2 copies of the acetyl-CoA, so another acetyl-CoA will go around the Krebs cycle. In total, you produce 4 carbon dioxide, 6 NADH, 2 ATPs, and 2 FADH2. So that's about the Krebs cycle. So it is where carbon dioxide is produced. So there are 2 of them here. But since there are 2 copies of acetyl-CoA, you multiply it by 2, so you have 4 carbon dioxide released. So interesting. You have a glucose with 6 carbon, 2 of which is released in the pyruvate oxidation, and 4 is released in the Krebs cycle. So that is where all of the carbon of the glucose goes become carbon dioxide. The GTP here later becomes ATP, and the FADH2 here is also produced. So we now know how many is produced in the citric acid cycle. So let's go back. In glycolysis, you produce 2 ATPs as a net and 2 NADH. In the pyruvate oxidation, you produce 2 NADH. In the citric acid cycle, it produces 2 ATPs, 6 NADH, and 2 FADH2. Now take note that the 2 ATPs over here and the 2 ATPs over here are produced, but the NADH and the FADH2 are all going towards the oxidative phosphorylation, and this is where the electron transport chain will come into the picture, so that the electrons that is carried by the FADH2 and the NADH will now be converted into ATPs. Do take note that in oxidative phosphorylation, 26 or 28 ATPs are produced, which would be the majority produced in the cellular respiration of just one molecule, whereas 2 ATP from citric acid cycle, 2 ATPs from glycolysis, with a grand total of 30 or 32 ATPs. Now here's a question, why is it 30 and 32? Well, depending where the 2 NADH will be going into from glycolysis, it can enter different types of shuttles, carriers, processes, so that it will produce differing amount of ATPs because of that. So we'll be discussing that a little bit later. Now finally, we're done with glycolysis, we're done with citric acid cycle. Let's now go to the oxidative phosphorylation. So what happens here? You will be noticing here that this is called the electron transport chain. It happens inside of the inner mitochondrial membrane, and then it's something that is related to the electron transport chain of the Calvin cycle inside of the chloroplast as well. So there's not much of a difference except for the enzymes. Now do take note that what happens here is that the NADH and the FADH2 will release the electrons that they are carrying. As the electrons goes through the electron transport chain, energy is released, and that energy is used in order to bring hydrogen ions into the other side of the membrane, going into the intermembrane space of the inner mitochondria. So what happens here? Since the electrons, as it carries through or goes through the ETC, will cause the hydrogen ions to go from here towards the inner space. So there will be an accumulation of a greater number of hydrogen ions here and lesser amount here. This causes a gradient. So we already know from diffusion and osmosis that particles will be moving from higher concentration towards the lower concentration. So the hydrogen ions will try to find a place where it can go through a hole, so to speak, so that it can go back towards the other side. And what is that hole? And that is where ATP synthase will be coming here. By the name itself, we know that ATP synthase produces ATP. How does it do it? Through the movement of H plus through the ATP synthase. Do take note that this is the electron transport chain. Important for H plus gradient to occur across the membrane. But for the ATP synthesis, where chemiosmosis will happen, because ATP synthesis is powered by the flow of hydrogen ions back into the membrane from higher concentration going through ATP synthase to lower concentration. As the hydrogen ion passes through, energy is released, causing the ADP to become ATP. And this is where 28 to 30 ATPs mostly are produced. So again, it is because of the accumulation of hydrogen on one side and the moving through the ATP synthase from higher concentration to lower concentration that produces ATP. So that's what oxidative phosphorylation will be all about. Okay, so let's now look at that grand total of ATPs produced. How did we get 30? How did we get 32 as a total over here? Now, first of all, we already know that there are 2 ATPs produced in glycolysis and 2 ATPs produced in the citric acid cycle. So that is where 4 of the ATPs out of the 30 and 32 will be coming from. So where did the remaining 28 to 30 comes from? It's through the NADH and FADH2. Now take note, in glycolysis, do produce 2 NADH. As it goes towards the electron transport chain, depending on whether it went through the malate aspartate shuttle or the other shuttle, the malate aspartate shuttle will cause it to be multiplied by 2.5, so the 2 NADH will produce 5 ATPs. But if it goes through the other shuttle, the 2 NADH is multiplied by 1.5, it only produces 3 ATP. So that is where the difference will be coming from. So the glycolysis, and as the NADH goes towards the oxidative phosphorylation, you have a total of either 5 ATP or 3 ATP at this stage. On the next stages, you have here the citric acid cycle. So aside from the 2 ATP, it produces 8 NADH and 2 FADH2. So what happens here? 8 NADH is multiplied by 2.5, you get 20 ATPs. FADH2 is multiplied by 1.5, you get a total of 3 ATP. So let's try to get the total. So you now have 20 plus 3, you have 23 ATPs for the whole of the citric acid cycle. Then you have addition of 2 ATPs, so 23, 24, 25, 26, 27. And then take note that the NADH will be producing, let's start with the 3 ATPs here. So 20 plus 3, 26, 27, 28, 29, 30. 20, let's do that. 20 plus 3, 23, plus 2, 25, plus 2, 27, and then plus 3 over here, it's a 30. So that's where the 30 will be coming from. If the NADH goes through the malate aspartate shuttle, it becomes 5 ATP, so it becomes 20 plus 20 plus 3 is 23, plus 2 ATPs over here, 25, plus 2 ATPs here, 22, 27, plus 5 ATPs, you will have 32 ATP, and that's where the 32 will be coming in. So that is how we compute the number ATPs produced, 32, 32. Okay. What is the difference of substrate level and oxidative phosphorylation? Well, you're already very familiar with this one. In substrate level phosphorylation, the phosphate to produce ATP comes from a substrate. Whereas in oxidative phosphorylation, the electrons moving through the ATC are the ones producing the ATP. And that's no different from, this can be further explained here. So the ATP produced here is from the glucose being converted to pyruvate. So there's a substrate. That's why it's called substrate level phosphorylation. On the other hand, there would be a lot of intermediate molecules here. The ATP are actually extracted from them, and thereby there's a substrate from which the ATP will be coming from, or the phosphate will be coming from. That's why citric acid cycle is substrate level phosphorylation. Glycolysis is substrate level phosphorylation as well. But the difference of the oxidative phosphorylation is that the FADH2 and the NADH will be electron carriers. Those are the one that produces the ATP as it goes through the ATP synthase, and not from any kind of substrate. Thereby, it is different from the substrate level phosphorylation. And that answers the question, what is the difference of substrate level oxidative phosphorylation? And this ends our discussion for this time.